Hello. So in previous screencasts, we have learned about carbohydrates and lipids and fats, and in this one we are going to quickly talk about proteins. Just like carbohydrates and just like lipids and fats, proteins are macromolecules, macro meaning large, and they are also going to contain carbon, hydrogen, oxygen, and nitrogen. Proteins are made up of molecules that we call amino acids. And if you remember of our study of functional groups, we learned about amino, which is a nitrogen and two hydrogens. So an amino acid will have an amino group, this NH2, and then on the other end it's going to have a carboxyl group. And carboxyl is C double bonded to an O, also bonded to another O, H. So let me just insert in a picture here to show you what this general structure of an amino acid looks like. Our amino group is shown here in green, N connected to an H and an H. Then over on the right hand side we have our carboxyl, C double bonded to O, OH. And so, as I said, proteins are chains of these amino acids. Specifically, we are going to have covalent bonds, which we call peptide bonds, that will link the amino acids together to form something called a polypeptide. So, a protein is a functional molecule that's built from one or more polypeptides. Proteins have a bunch of different uses. Some will control the rate of reactions, and they can help to regulate processes that happen in cells. Other proteins are important for cellular structures, and other proteins will transport things in and out of cells, or they can help to fight disease. So there's a peptide bond that's going to connect the amino group of one amino acid to the carboxyl group of another amino acid. So we're specifically talking about this portion being able to combine with this portion on a different amino acid. You can see we put a peptide bond between there, but another key thing is shown right here, which is that a water molecule will be released when this peptide bond is formed. Also note that between alanine and serine here, this molecule is basically the same as this molecule. The only thing that changes is this purple portion, and this is what we call the R group section of the molecule, and this is just what will distinguish one amino acid from another, and it will vary depending on what the amino acid is. Now for some more information about the structure and function of proteins and amino acids. There are more than 20 different amino acids that are found in nature, and as I said, all amino acids are going to be identical in the regions where they're joined together by those covalent bonds, and so they're very uniform, and it allows basically any amino acid to join to another amino acid. Again, that's by having the amino group of one combined with the carboxyl group of another. And though these look very similar, they are some of the most diverse molecules that we have, and it's because of this R group shown in purple. And the different R groups will have a really big range of different properties. Some are acidic, and some are basic, some are polar, some are nonpolar, and some may even have really big ring structures in this area. So proteins have different levels of organization. We know that their building blocks are amino acids. Here's an example of one, and another example of an amino acid. And those amino acids we put together into what we call polypeptide chains, and that's according to instructions that we find in the DNA. So to help understand the larger molecules, scientists describe proteins as having 4 different levels of structure. A protein's primary structure is going to be the sequence of its amino acids. its secondary structure, then we have the folding or the coiling of that polypeptide chain. Then there's the tertiary structure, which is going to be the complete three-dimensional arrangement of the polypeptide chain. And if proteins have more than one chain, then we often say that they have a fourth level of structure, which is just going to describe the way that the different polypeptides are arranged with respect to each other. This is called the quaternary level. So let me show you a picture here showing these different levels of the protein structure. This is showing the 4 levels of structure in hemoglobin, which is a protein that you find in red blood cells, and its job is to bring oxygen around in the bloodstream. So the shape of the protein will be maintained by a bunch of different forces. We have ionic and covalent bonds, there's hydrogen bonds. We'll learn more about its shape in a little bit. But again, there are 4 subunits in this protein hemoglobin, and you can see that there's this heme group right here, which contains iron, and it gives hemoglobin its reddish color. And then we have an oxygen molecule that will bind tightly to each heme molecule. So that's some basic information about proteins. Keep in mind that we are often going to be talking about enzymes, which are a type of protein. And if you recall from chemistry, enzymes are things that change the speed of a reaction. Something to keep in mind about enzymes that's important is that enzymes are very specific, which means that each enzyme will only work with one reaction or one small group of closely related reactions. So for an enzyme to work, we need something called a substrate to bind to it, and they have to fit together perfectly. Every enzyme is going to have something called an active site. So this is our enzyme, and it's this location here where our substrate is combining. And we call this the lock and key, because it's like here is our lock and this is the key that will unlock the door. And make sure you know what I'm talking about when I say that this is our active site. So the active site is just an area on the protein surface where the substrate will bind. The shape will fit the substrate, just like a glove on a hand or a lock fitting into a key. Clearly, only substrates with a certain shape will have any chance to bind effectively in this example. We call this the lock and key hypothesis, but it doesn't give really the whole picture because enzymes are a little bit more subtle than this. It's not enough for a substrate to just fit into the active site. It also is going to need some attractive forces to help keep it in that place. Once the substrate and the enzyme have bound together, they will together form what we call an enzyme-substrate complex. But the idea that the active site has a fixed shape is not exactly accurate, and what we've now come up with is we use something called the induced fit model, which is where the walls of the active site cleft, they're going to move a little to wrap around the substrate as it binds. And so this flexing will put the active site and the bonds in the substrate under strain, which weakens the bonds and actually helps to lower something called the activation energy that we need for the reaction. You can see here that this active site is not the same shape here on the enzyme when the substrate is in it. So this enzyme will change shape to fit the substrate and kind of hug it tightly. So this induced fit model is they came up with this after they proposed the lock and key model, right? If going back to there, here we see an example of the lock and key model, which just states that our substrate acts like a key to the lock of the active site, and then the active site and the substrate are exact matches for each other, just like puzzle pieces fitting into one another. And with this model, you can only have one particular substrate that's a precise match for the enzyme. And once the enzyme finds its exact counterpart, then the chemical reaction can begin. But in the induced fit model, this is considered the more generally the more correct version, which is just saying that the active site and the substrate are initially not perfect matches for each other, but rather the substrate is going to induce a change in the shape of the enzyme. And so it's just like placing your hand in a glove. You get your first finger in, it may be a little difficult, but once you get that first step in, the glove will slide on easily, with less energy because it's now properly aligned with your hand. So that's all I have to tell you about the induced fit model. Don't worry too much about this Koechlin theory. This is the guy that made it up, but again, the substrate is inducing a change in the enzyme. It's making it change its shape so that it can fit perfectly into the active site. And that's it for this screencast. Bye!